Students, it is second video on O Levels Biology 5090, Chapter 1. We were discussing cells in part 1. Today, the part number 2, in which we are moving towards the another organelle, which is only present in plants, that is chloroplast. And chloroplast is only present in plants. It is absent in animals. It has a green pigment. It has a green pigment, which is chlorophyll. It gives green color to the plants. So green plants are green because of this chlorophyll. And its function, very important function of this chloroplast or chlorophyll is to capture the sunlight for the process of photosynthesis. That is, the photosynthesis cannot take place without chlorophyll or chloroplast. If they are absent, it means the photosynthesis will not take place. Now, I want to show you the diagram of this chloroplast in the next slide. And in the next slide, you can see the different parts of the chloroplast. As you know, the students, in the last part, we had discussed about mitochondria, which has the double membrane structure, and same is the case with this chloroplast. It also has double membrane structure, which is outer membrane and inner membrane. And beside this outer and inner membrane, there is membranous structure, which is thylakoids. And you can see the very important part, which is stacks of thylakoids. Stacks of thylakoid, they are called as granum. And inside the granum, the center of this chloroplast is filled with stroma. So this is all about the structure of chloroplast, but we want to know that its actual function is to absorb the sunlight for the process of photosynthesis. So it is very important in the plant cells. Now the next, another important organelle is ribosome. Ribosome is very complex molecule, actually just you can say play a part in protein synthesis. So we will say that is a complex molecular machine found inside the living cells. Living cells mean in both plants and animals that produce proteins from amino acids during the process called protein synthesis. And you know, the protein synthesis is very important part which is taking place inside the living cells, whether they are animals or plants. So ribosome has very important function. Now I would like to move toward the next. You can see the ribosomes are, you can say this is an animal cell, and you can see the ribosomes are located not only in cytoplasm, they are also present on the endoplasmic reticulum. They are also attached on the endoplasmic reticulum, and endoplasmic reticulum is actually the transportation system inside the cell. So ribosomes are freely available in the cytoplasm as well as they are present on the endoplasmic reticulum. But again, their function is the same, that is, they are involved in protein synthesis. Next, you can see a single ribosome present on the screen. The ribosome size is 10 nanometer, about 10 nanometer, and it consists of two subunits, large subunit and small subunit. And you can say the instructions to make protein on a messenger RNA molecule in the nucleus, it is present over there. So in this way, I would like to say, I will not go in detail because according to the syllabus, we must know what is ribosome, what is its function. So we will not go in detail at O levels. Now, next, I would like to discuss about the bacterial cell. We had discussed about animal cell, plant cells, different organelles, and their functions as well. So bacterial cell, basic components of a bacterial cell include, now you can see the bacterial cell also has some organelles and parts of it. Cell wall is present, cell membrane is also present, cytoplasm is present, Ribosomes is present, nucleolide which contain DNA or RNA. Some bacteria also have flagella and villi. Flagella for the movement from one place to the other, and villi to attach it with some objects, and locomotion and adhesion. Flagella for locomotion and villi for adhesion. The bacterial cell provides shape and rigidity. The bacterial cell wall provides shape and rigidity. This is the basic function of the cell wall, which gives shapes, and it gives rigidity to the cells, protecting bacteria from mechanical and osmotic stress. So it also helps in protection. So now I would like to move towards the next slide, in which I will show the structure of the bacteria. So here you can see the diagram of the bacteria, in which I had discussed in the last slide that it has cell wall, it has cell membrane, it has ribosomes, cytoplasm, and gelatin-like capsule, which is uh, for protection in adverse environment. And flagellum is for the movement, and these, you can say, the cilia. And also you can see there is no proper nuclear membrane around it. So it don't have proper nucleus. So it means it don't have proper nucleus. It has DNA. It has DNA inside in two forms. One in this form, and second, it also has a circular DNA which is called as plasmid. So it don't have proper cell membrane around the nucleus, or nuclear membrane around the nucleus. So it is called as prokaryote. Bacteria is called as prokaryote due to the absence of nuclear membrane, because it don't have a proper nucleus. Now I would like to move towards the next slide. And in next slide, we will discuss the differences between an animal and plant cell. And students, you have discussed clearly about all organelles of the animals and plant cells. Now, I would like to say that animal cells, they are smaller in size, whereas the plant cells are larger in size. The size difference, cell wall is absent in animal cell, whereas it is present in plant cells, which gives the plants rigid and shape and protects the plant cell. Many small vacuoles are present which are temporary, but a single large vacuole is present which is permanent. Chloroplasts are absent because animals are not making any type of carbohydrates inside, or they are not involved in the process of photosynthesis. They depend on ready-made food which is manufactured by plants. So plants have chloroplast uh, and nucleus in the center because they don't have a large vacuole. So nucleus is on one side, and centrioles are present and centrioles are absent. And what are centrioles? Centrioles are those parts, or you can say the part of the cell which are involved in cell division. They are helpful at the time of cell division. They are absent in plant cells, but the plant cells divide without any help of these centrioles. The cell division can take place without these centrioles. Now I would like to move towards the next topic, which is magnification. Magnification is actually the action of magnifying something or process of being magnified. So magnification is a very simple term which we use in biology, uh, with the help of which we calculate, with the help of our drawing, we calculate the magnification of the diagrams or pictures which are given. Magnification is the process of enlarging the apparent size not physical size of something, apparent size, not physical size. So I would like to move towards the next slide, in which I will show you the formula of magnification, and then we will try to calculate the magnification. So the formula for magnification is the size of the image divided by actual size of the object. Size of image and actual size of the object, image or drawing, which you will draw. So magnification is size of the image divided by actual size of the object. And if you want to know the actual size of the object, then you can know the size of the image divided by magnification. So you have to learn these formulae to find out the process of magnification. And you know, students in O level, there is very simple magnification. And the method of this to know the magnification of the object is here. I would like to show you. This is the actual diagram which they have given. Let me. So this is the actual diagram. You have to write in this way. You have to measure this. It is around 1.5 centimeter. A size of this diagram, actual diagram, is 1.5 centimeter. Now we have to draw the larger than this or double of this. So you know the drawing. So in this way, you have to again, with the help of, you have to do this with the help of scale, not just like me, because I'm drawing it by without the help of any scale. So in this way, you can have, let me, yes, if we measure it, students, keep in mind, there should be a straight line. It should be a straight line, not just like me as I have drawn it. Now you can see that it is about 4 centimeter. So magnification is size of the image over actual size. So first of all, we have to change the centimeter into millimeter. 1.5 centimeter is equal to 1.5 multiplied by 10 is equal to 15 millimeter. So 4 into 10 is equal to 400 millimeter. So here you can see that we are going to, this is the image size, which is 400 divided by the actual, which is 15. So we will calculate 400 divided by 15. It is around 26.6 or 27. So magnification is 27. So this is the actual way to find out or to calculate the linear magnification or the magnification of the simple diagrams. Students, today we have completed our part 2. Inshallah, we will meet in the next part and we will complete this topic cells. Thanks for watching me. Allah Hafiz.